This one's going to incorporate C3 and C4 together because they both go together really nicely. So C3 is basically the I might. C3 is the movement across the membrane, and C4 is the why do we need it, basically. Okay. So C3 is movement across the membrane. C4 is why do we need that? And so we'll probably cover that one first. So we have, let me just see if I can get rid of that. Okay, so we've got our membrane. We always draw it like this. The phospholipid membrane, phosphoheads hydrophilic, the tails hydrophobic, and they separate the inside from the outside of the cell. Nick, why do we need to have that separation?
So that
this is live. People are streaming this on YouTube world as we speak.
I'm not sure.
Anybody? We've got this membrane. It separates the outside from the inside, but why do we need that? Why do we want that? Reese?
To separate the inside of the cell's contents with the outside.
Excellent. So we've got things inside the cell that we don't want to leave. We've got vacuoles, we've got mitochondria, we've got maybe chloroplasts, we've got our nucleus. We can't have all that stuff just floating around in space. We need that to be enclosed. So that's one thing, to maintain our structure and to add our organelles to make sure that they're in there and they can do their job. But there's other things that we need as well. What else would we need different inside than outside? And to give you a hint, why do we breathe? Why do we eat? They could give you some clues. Energy. So we need energy inside our cells. The cells don't get that directly. Well, they get that directly from the outside environment, but it needs to get to that outside environment in some way. So we eat molecules, we digest them, we put that in the extracellular fluid, and then we need to incorporate it into the cell. Okay? So we need energy going in, and that energy is in the form of glucose. What else? What things might we need to get out? Wastes. So we want waste going out. Okay. We need things like O2 involved in energy coming in, and we need things like CO2 leaving. Okay. So basically inside the cell, we need to control lots of ions. So we've got inside the cell sodium ions different inside to outside. So I've got about 15 millimoles inside compared to 145 millimoles outside of sodium. Okay. We need potassium at different concentrations inside and out. Magnesium. Calcium. We need water to be controlled. And we need some of those other things like oxygen and glucose like we've already mentioned. So we need those at different concentrations inside compared to outside. These two here are used in nerve signals, and calcium is used in muscle contraction. So we need to make sure they're perfect, otherwise the cell might die. Okay, so the internal concentration needs to be different than the external concentration, otherwise things go wrong. Yes, Matt?
How does magnesium stop man?
Magnesium is probably involved in the control of muscle contractions in some way. The one that I know more in detail is calcium, and glucose. So muscle contractions are required. Basically, you have this exchange between sodium, potassium for the electronic signal, and then you have ATP controlling the uptake of calcium. So when you have a muscle contraction, you release calcium, and that signals your muscle to contract. If you don't have ATP, you don't have sugar, then there's nothing that reuptakes that calcium, so you're in this constant state of contraction. So magnesium's obviously involved in that somehow as well. But basically, these are why we have sports drinks and things like that, to make sure that our balance of these ions are perfect, except they don't really work that well, the sports drinks. We also need things like hormones to be controlled, but we need to stop things from coming in. This membrane needs to be able to select what molecules can come in and what molecules are prevented from coming in. So there's some things that we'll look at about the nature of that allows that to happen. So we need to be able to allow hormones to come in. We also need to be able to signal between adjacent cells, and all that needs to happen very efficiently. Okay? So what we end up having... Oh, this has changed around a little bit. What we end up having is ability to move things across that membrane. So we have several processes. They can be classified into two main areas. Does anyone know what they are?
Passive and active.
Beautiful, Nick. Redeemed yourself.
Oh, no, thank you.
Okay, so we've got passive and active. Okay? So these are our two types of transport that a cell will employ to move things across the membrane. So what does passive mean?
No energy required.
Okay, no energy. And it's true, because we do have our free energy, we have the kinetic energy of the molecules, so there is energy there, otherwise nothing would work, but we're not having to provide any additional energy in the form of ATP. Okay. Whereas our active transport requires ATP. Now, if you did Year 11 biology, some of these you would know, others you might need to brush up on. Okay. Passive. We have three main ones. Diffusion. Facilitated diffusion. And osmosis. Okay? So these are the three that we require, or these are the three that require no energy. Okay? So we'll go through each of these in detail in a second. And then we have, let me count, 4 active processes. Well, really 3 with a couple of sub-branches. We have one that just sort of gets called active transport. Oops. So we've got active transport, which is using a protein pump. So these are the ones that are going to pump ions across. So these are active. We've got endocytosis and we've got exocytosis. And under endocytosis we've got phagocytosis and penocytosis. Okay. Now these passive and active processes need or don't need energy because of one thing. Okay? These passive processes don't need energy because why? Gradient. Because of the concentration gradient. So these all move. Osmosis is slightly different, but these all move with Okay, their concentration gradient. These here are moving against their concentration gradient. Okay, so we're moving against, that requires energy. Okay, if we need the production of a vesicle, we need to move that vesicle using motor proteins, we require energy. To fuse it with the membrane and to expel those contents or take them in requires energy. So if we're pumping ions against their concentration gradient, so we're having to actively do that all the time, that requires energy. Okay, and that energy comes in the format of ATP, the currency of the cell. We will start with diffusion. Okay, so if we look at diffusion, diffusion describes the movement of any molecule based on the kinetic energy that it has, which is just the vibration of molecules. This is a form of heat energy, so the more heat the molecules have, the faster they will move, but it's a random process. So if we just take a container, okay, and we put some molecules inside, and we put them next to each other, those molecules are going to be moving. And molecules are going to move in straight lines. And they're going to move in a straight line until they collide with something. Okay, so it's very straightforward. The reason why they move outwards is because that's where there's space. Where there's space, there's less collisions, so they'll move out in that direction. Okay, so if we follow the path of one molecule, it will move in this direction, collide, move in that direction, and spread out. This molecule here might move here, collide, and then move off in this direction. Here, collide, move off, collide, move off, collide, move off, until eventually, if we draw our container again, those molecules are just moving around in a uniform way, and they'll continually do that. So they're equally spread out. They've reached an equilibrium where there's an equal distance between them. The likelihood of collision is at now a steady rate, and so they're just going to be moving around. Okay, so they've gone from an area of high concentration, so this is high, to an area of low concentration. And that's just a random process. So we write the definition for this as, let me see if I can do it in text. The random movement of molecules from high concentration to low concentration until equilibrium is reached. Oh. Okay, and I'll just move that around. Okay, so this will occur for all molecules. They'll have that kinetic energy, they'll be moving, they'll be colliding. So over time, they'll be moving from an area of high concentration to low concentration. The thing that sets up that concentration gradient is again our cell membrane. So our cell membrane is going to have molecules maybe high on this side, low on this side, so we're going to get movement across the membrane, provided it can go over that membrane. If it can't pass that membrane, then it's a localized environment, and therefore we're only going to reach equilibrium here. Okay? Once they can pass, then it's a difference between these two. And you remember that animation that I showed you shows that quite clearly. This will occur with water. This will occur with any molecule. The difference with water is that sometimes it then interacts with molecules on the other side, preventing it from coming back. And that's when we get our osmotic effect. Okay. Next is facilitated diffusion. Okay, facilitated is very similar. We have our membrane. We have outside and we have inside. Now this membrane is very selective. Okay, it doesn't just allow anything through, thankfully, like our bouncers at a nightclub. You need to wear enclosed shoes, no exposed tattoos, no provocative clothing, or maybe they promote that, I'm not sure. But it is selective, okay? So in our cell membrane, it is selective for polar molecules. So polar, non-polar molecules or charged molecules have trouble getting through. So charged molecules have trouble getting through. Water molecules can't get through because they get trapped in the middle of the membrane where it's hydrophobic, okay, because of their polar nature. We have large molecules. Molecules which are just too big to pass through the membrane, to pass through the gaps in the membrane. So if you're a molecule and you're any one of these things, you can't get through the membrane regardless of your concentration gradient. You need something to help you. That's where facilitated diffusion comes in. Facilitated diffusion uses proteins embedded in the membrane. Okay, and these are... Okay, so we have our membrane. We want to control the water movement in the cell, but we know water movement is a passive process. We can't actually control it. The water, Matthew, follows the solute concentration. So if you want to control water, you have to control the solute. So as you all correctly identified, if we can make an area with, let's say, high solute compared to low solute, then our net movement of water is going to move in this direction. Conversely, if we switch those solute concentrations around, the net movement of water is going to move in the other direction. Okay? Provided our solute isn't permeable to the membrane, or we do something else, but we'll get to that in a sec. So that's all well and good. Control the solute concentration, control the solute concentration. But how? Reese?
If you have a low solid concentration inside the cell and high outside, if you want high inside, we'll just take away the high solid concentration from outside the cell.
How?
Using that exchange process.
What exchange process?
Selective exchange.
Excellent, Nick.
Thank you.
So what we need to do is we need to change that concentration. As Reese mentioned, we need to make one different to the other, and that way we can control the movement of water. The way that we do that is by incorporating our pump. So this protein pump is going to, if we want salt to be high on this side and low on that side, in this instance. In fact, in a cell it's actually the opposite, but we'll go with this way. We are going to pump sodium ions from outside we're going to pump sodium ions into the cell. Okay, we're going to pump them in. By pumping them in, we now have this solute concentration difference. The water molecule is going to move across, interact with that salt that's becoming now higher on this side. So the water molecules are going to interact with those salt molecules, and then they're not going to be able to come back. If we want to now change that, we stop pumping, or we then pump them back out. Okay, there's a very famous sodium-potassium pump that takes out 3 sodium and brings in 2 potassium, and that changes not only the concentration of those ions, but as you can see, the charge as well, because we actually alter the charge, and that's how nerves work. So to control osmosis, we need to control the salt, because we can't directly move water. We don't have a water pump, okay, like you can get from Bunnings. Okay, so we have to move the salt. Okay, so just to recap osmosis. Water molecules move through diffusion processes. It's a random movement. It goes from high to low until equilibrium. Indicated by this diagram here. And I've incorporated a laser. I've got all the tools. Okay? If, however, we have a situation set up where we've got a membrane that is permeable to water, so allows water to go through, but is not permeable to, say, a solute like salt, then the water will move across through its random diffusion process. It will interact with those salt molecules and join to it. Because those salt molecules can't pass back through the membrane, the water becomes stuck on that side and can't move back. Over time, that water molecules will move across. We'll have a net movement of water across. You'll still have some moving back, but overall the water molecules will move across, get attached, and then we'll have the water level increase on that side and decrease on that side. So that's the gist of osmosis. Okay? Controlled by concentration. If we want water to move across faster, we have a larger concentration gradient. If we want water to move slower, we have a slower concentration gradient, a smaller concentration gradient. This becomes very important to a cell. If a cell inside has a really high salt concentration, then lots of water molecules will move in, expand, and eventually pop, explosion, water everywhere, cell everywhere, disaster. Likewise, if we have a really high, Nick, people might experience this when they, in the ocean, when water, maybe in the bath, you might experience this as well, a bit of shriveling, okay? When you have a bath with lots of salts, maybe bath salts, that salt concentration is higher than the water molecules inside your cells. Water moves out, shriveling up, and you get that sort of wrinkly appearance on your skin, and it shrivels up, okay? That's really bad for cells as well, especially animal cells and plant cells. This is why plants, flowers wilt. It's because water has moved out of them. You want to get them standing tall again, you put them in water, water moves back in, they stand more erect. Yes, Shana? I don't know. You should find out for us. Okay, our first active process. Now, we just spoke about it briefly in the movement of water, because that's how we actually control the salt concentration, is we use a protein pump. Okay, so here's our membrane. We have our protein pump that is basically transverses across the membrane, or is the whole way across. That protein pump is specific to a molecule or a 2-molecule exchange, and it will move molecules, and it has a cost, and that cost is ATP. So every time we want to move molecules across, ding, we have to pay a fare of an ATP molecule. And the easy way, in an exam, the types of questions that you'll get is you won't have that ATP there, so that won't be there. What you'll see is molecules. I'm going to draw them in funky green. Okay, so you'll see molecules that look like this. And the question will say, is that movement of that molecule active or passive? How can you tell?
Because when the more ATP is.
No ATP is listed.
Yeah.
You can't. There's not enough information there for you to tell because you need a direction of movement. So, if I give you a direction, now you'll be able to determine whether that process requires energy or not based on very simple observation. So if I draw the arrow like this, indicating that the molecules are moving from this side to this side, you can then determine whether that's an active or passive transport. Now, is that active or passive? Active. Okay, because we're moving against the concentration gradient. This is low, this is high. Because we're forcing molecules to where they wouldn't necessarily go through a random movement, it requires energy. We need ATP, which means it's active process. If the arrow was going in this direction, it would mean it's diffusion. If the arrow was going through the channel, it's now facilitated diffusion. So you need to be able to distinguish between those three options. Is that diffusion? Is that facilitated diffusion? Is that active transport through a protein pump? Or if it's just the movement of water molecules, is it osmosis? Okay, so active transport is simply moving molecules against their concentration gradient using energy to do so. Okay, so that is active transport, protein pump. Okay, that's that one done. Easy. Next is our, let's say, exocytosis. Okay, now last lesson, when we looked at the membrane, we realised that everything inside the cell that is enclosed, that is like a package or a sac or something, is made up of phospholipids. Everything. The nuclear membrane, phospholipids. Endoplasmic reticulum, phospholipids. Golgi apparatus, or the Golgi apparatus, phospholipids. Vacuoles, phospholipids. Vesicles, phospholipids. Everything is made up of phospholipids. And that's awesome because it means that they can interact with each other and they can fuse together or separate from one another, and that allows some excellent things to occur. So if I have a cell, exocytosis means a vesicle leaving or expelling its contents out of the cell. So what it looks like, we have our vesicle. It moves towards the cell. It moves towards the cell. And then, if we join up, the vesicle now fuses with the membrane because they're both phospholipids. The membrane then pulls apart. Oops. The membrane. Can I erase that? Oh, I can't. I'm going to try. The membrane then disappears. Okay, it's not that it's disappearing, it's just combining with the vesicle, and then we can have our contents released. Okay. Vesicle moved towards the cell membrane, and now we're watching that. When they watch that video, we can see that's probably motor proteins moving them along the cytoskeleton towards the outer membrane. Fuses to the membrane, membrane opens up, contents are released. Exo means exit. Exocytosis. Yes, Reese?
Once the vacuole, once it's expelled, its contents.
It will just then merge in and just become the cell membrane. Okay, because remember it's made of the same stuff.
Yeah, I didn't know if it was back in.
Not necessarily, but it could wait there And because the process in reverse, going back in,
That causes a disease. Like, say if something got in there and it's such before it causes
Does it contain
okay, listening up, thank you. So remember, exiting. We only have one form of exocytosis. Okay, it just is basically for anything. Any vesicle binding, fusing with the cell membrane, expelling its contents is exocytosis. Endocytosis, think of it in the opposite. The membrane folds in, creating a circular void that things can move into. If it is a large solid piece of material, maybe like a piece of bacteria or bacteria itself, we call it phagocytosis. Okay, basically means eating. And if it's, so eating, if it's pinocytosis, it means taking in dissolved material. Okay. And that's it. They're our transport mechanisms, all of them. Okay, our three active: diffusion, facilitated diffusion, osmosis, and then our active transport protein pump, exocytosis, endocytosis. And that is it.